Good day, everyone. My name is Angelica Rachel Carpio, and my thesis advisor is Dr. Lemel Tayo. And my thesis is entitled with the role of cyanidine 3-O-glucoside and vaccine metabolites in modulating HSP90AA1 and PTGS2 in gastric carcinogenesis. For the introduction, number one, gastric cancer is the fifth most common cancer and the fourth leading cause of cancer-related deaths worldwide. And this predominantly affects older adults. And number two, despite advances in treatment like surgery, chemotherapy, radiation, outcomes for advanced GC still remains poor. And this is largely attributed to late-stage diagnosis, aggressive tumor biology, high risk of metastasis, and the development of resistance to conventional treatments. So, therefore, there's a growing interest in complementary therapies, particularly for plant-derived compounds due to their lower toxicity and bioactive properties. So, this highlights the urgent need for more less toxic and targeted plant-derived compounds. Now let me introduce to you to the Vaccinium genus. The Vaccinium genus comprises berry-producing plants. Some of this is the northern high-bush blueberry, rabbit-eye blueberry, low-bush blueberry, bilberry, cranberry, and lingonberry. And this genus actually comprises 450 species. The Vaccinium metabolites, on the other hand, are recognized as a significant supplier of bioactive compounds, as it has high content of antioxidants, has anti-inflammatory agents, and has anti-carcinogenic effects. Now that we know that Vaccinium metabolites have been linked to reduce inflammation, improve immune responses, and protection against various cancer, despite all their general health benefits, there is still limited research into their specific role in gastric cancer prevention and treatment. So there is a lack of studies on how Vaccinium metabolites interact with cancer-related pathways, especially in gastric cancer. So therefore, there is a need for a deeper understanding of how these bioactive compounds can influence gastric cancer progression. Now, for the objectives of my paper is number one, to identify the key upregulated genes and pathways in gastric cancer using RNA sequencing, and number two, to identify the essential genes and pathways using network pharmacology, and number three, to validate the binding affinity of Vaccinium metabolites to gastric cancer-related proteins. So the hypothesis of my paper is that Vaccinium metabolites may inhibit gastric cancer progression by modulating key cancer-related pathways that offers a novel therapeutic approach for gastric cancer prevention and treatment. The significance of my paper is to demonstrate the potential for Vaccinium metabolites as candidates for gastric cancer therapy and to establish a computational foundation for targeting specific genes and pathways. For the methodology part, number one is the collection, screening, and ADMET-based filtering of metabolites in the Vaccinium genus. In here, the metabolites present in the Vaccinium genus were screened and their canonical smells were retrieved, and then these were entered to ADMET Lab 3.0 to filter them based on Lipinski's rule, Pfizer's rule, and cell permeability. After that is the target gene prediction in the Vaccinium genus. This is where the canonical smells were again entered to superpred to predict their target genes for Vaccinium genus based on their probability scores. And after that is the collection of RNA sequence data associated with gastric cancer. And here, their RNA sequence data were retrieved and pre-processed using Recount3, our library. And then the data included gastric tissue samples from various stages ranging from gastritis to advanced gastric cancer. And after pre-processing, VE-SEC2 was used to identify differentially expressed genes for each condition with upregulated and downregulated genes. Then after that is the compound and protein-protein interaction network construction analysis. And here, CPI network was created to visualize the relationships between compounds and their targets, and the gene lists per cluster were analyzed used for PPI through StringDB, and the hub genes were identified by degree. And then after that is the gene ontology and KEGG analysis, wherein These pathway enrichment analyses were performed to explore their biological mechanisms through which the vaccinia metabolites affect gastric cancer. So, the common genes that were identified from vaccinia metabolites through superprediction and gastric cancer RNA sequencing data were processed using David. And finally is the molecular docking. Molecular docking was employed to evaluate the binding interaction between the target compounds from the Vaccinium genus and the hub target genes identified in the study. So, the docking simulations were performed using CBDoc2. Now, for the results and discussion, these are the 5 key bioactive anti-cancer metabolites from the Vaccinium genus. This includes resveratrol, ursolic acids, scopolamine, catechin, and cyanidine PO-glucoside. So these 5 key metabolites were selected based on Lipinski's rule and cell permeability. Lipinski's rule were included since the Lipinski's rule suggests that there is a higher likelihood of suitability for oral administration and potential effectiveness in drug development, and additionally, cell permeability values greater than negative 5.15 were selected since this indicates their potential to effectively transverse intestinal cell membranes through passive diffusion, carrier-mediated uptake, or active transport. This is the compound protein interaction maps for each stage in Korea's cascade based on accepted vaccinia metabolites and their predicted targets. So this CPI networks visualize how the vaccinia metabolites interact with protein targets involved in gastric carcinogenesis. This is the model for the RNA sequencing data that shows the histological progression towards gastric carcinogenesis. And interestingly, this is aligned with the Korea's cascade model. So our data starts with the early stage gastritis. So this starts with cluster 2 chronic gastritis where there is an inflammation of the stomach lining, and this can be caused by infection with Helicobacter pylori or maybe an autoimmune disorder. After that is the atrophy wherein there's a thinning of the stomach lining. Then after that is the metaplasia where there's a replacement of the mature cell type into another mature cell type, and this is still considered as benign and reversible. And after that is the tumor progression in cluster 1. This is where dysplasia starts. So this is the replacement of the one mature cell type into another less mature cell type. And then this is considered as precancerous and non-reversible. And then after that is the early gastric cancer and the primary tumor. Now this is the top hub genes from PPI. We have MMP9. So this is known to break the extracellular matrix that regulates the cell behavior. We have PTGS2 or the COX2. The cyclooxygenase 2 is known to produce prostaglandins that causes inflammation. We have HIF1A, which is a transcription factor that regulates cellular responses to hypoxia or low oxygen. ERBB2, which is a receptor tyrosine kinase that belongs to the AGFR family. We have the HSP90A1, which stabilizes the EGFR and AKT pathway that promotes tumor growth, and the other genes from cluster 2. So, all of these genes, the overexpression of these genes rather, leads to tumor progression, apoptosis, and metastasis. However, this paper only focuses on the cluster 1, which is the early stage gastritis. Next is the gene ontology and KEGG enrichment analysis. So in here, in the gene ontology, this reveals the functional implications of the intersecting genes that provides us the insights into their biological roles and potential as therapeutic agents in gastric cancer. So we can see in the biological process, we have the protein phosphorylation and positive regulation of cell migration emerge as the highly enriched. So this indicates their crucial role in signaling and cancer progression. Then after that, in the cellular component, we can see that the cell surface appear to be the highest fold enrichment. So this reflects the importance of the membrane-bound processes in cancer-related mechanisms. And for molecular function, we can see that the kinase activity and the ATP binding were significantly enriched. So this suggests that their role in phosphorylation and processes and cancer signaling pathways. In the KEGG pathway analysis, on the other hand, this is to map the intersecting genes to known biological pathways. So this enhances our understanding of their roles and in cancer-related mechanisms. So we can see that the pathways in cancer appear to have the highest counts. So this represents the interconnected mechanism driving tumorigenesis. And then next is the microRNAs in cancer appear to be the second highest count. So this highlights the regulatory role of microRNAs in gene expression and cancer development. This is the heat map of the molecular docking results of the key metabolites from the Vaccinium genus and central target genes. So for the proteins from gastric cancer, we have ERBB2, HIF1A, HSP90A1, MMP9, and PTGS2. And for our ligands from the metabolites of the Vaccinium genus, we have ursolic acid, scopolamine, resveratrol, cyanidin 3-O-glucoside, and catechin. So this is to evaluate the binding interaction or their binding energies. So the values ranges from -6 to -11.1. So we can observe that they all have stable binding energies. Finally, this is the top 3 ligand receptor complexes. First, we have the HSP90A1 cyanidin 3-O-glucoside complex that has -11.1 binding affinity. So as HSP90A1 is known for protein folding and cellular homeostasis, this is the highest observed binding energy. So this suggests that the cyanidin 3-O-glucoside can actually disrupt the chaperone function and can also destabilize oncoproteins that eventually inhibits cancer proliferation. And then we have PTGS2 cyanidin 3-O-glucoside complex. It has -10.1 binding affinity. So as COX-2 inhibitors have anti-tumor effects, this can modulate PTGS2 activity, and this can also reduce inflammation and cancer cell survival by disrupting COX-2 mediated pathways. And then lastly, we have ERBB2 or ursolic acid complex that has -9.7 binding affinity. So the overexpression of the ERBB2 can actually be inhibited by ursolic acid, and this can also induce apoptosis and disrupt the cell cycle progression. In conclusion, key therapeutic metabolites were identified. So C3G or ursolic acid, resveratrol, scopolamine, and catechin as the promising bioactive metabolites from the Vaccinium species with potential therapeutic effects in gastric cancer. And then the targeted molecular interactions, C3G demonstrated the ability to inhibit cancer-related targets such as the HSP90A1 and PTGS2. Also, ursolic acid showed a potential to inhibit ERBB2, which is involved in cancer proliferation and metastasis. And then lastly, the relevance to gastric carcinogenesis. So the RNA sequencing data results aligns with the Curious Cascade model that elucidates the intervention points for the early and advanced stages of gastric cancer's progression. Ultimately, this study lays the groundwork for leveraging natural metabolites from the Vaccinium genus in gastric cancer prevention and treatment that connects computational insights with experimental possibilities. Moreover, it is recommended for experimental studies like in vitro and in vivo to confirm interactions and therapeutic effects. Also, structural and chemical specifics of the binding interactions should be studied. And lastly, addressing challenges like bioavailability to transition towards to drug development is also recommended. Again, I'm Angelica Rachel Carpio, and thank you for listening.